Kia ora everyone, welcome to the next mini lecture, and we're going to continue to talk about endophytes here, and we're going to continue to talk about the analysis of experimental data. And what I'm going to do in today's mini lecture, or in this mini lecture, is look now at a real example of an experiment that scientists did to assess whether endophytes give protection against a fungal pathogen. So we'll have a look at what they did and see if we can interpret their data. So I'll turn my video off and we'll get started with going through the slides. Okay, so again, the idea here is to see whether endophytes protect their host from a plant pathogen, and that's a fungal plant pathogen. And so what we're going to do now is look at a real experiment where that experiment was designed to test this. So let me introduce the experiment. And here they're looking at different endophytes, so not class 1 endophytes. In this case, they're class 3 endophytes. And they used 4 different species of class 3 endophyte that we'll look at when we look at the results. And they inoculated these class 3 endophytes into 6 different poplar species. So these poplars are a kind of tree from the genus Populus. And so that was their experimental system. So in this case, they're using trees. And their fungal pathogen that they used was something that's called Melampsora. And this is a rust pathogen. So rust pathogens are pathogens you might have heard of that affect a wide variety of plants. They're famous for being devastating crop pathogens. And rusts come from a group of Basidiomycetes. So they're a Basidiomycete pathogen. And you may learn more about rusts later on in this course. And so they were interested to see whether these endophytes inoculated into these trees provided protection against this Basidiomycete pathogen, rust pathogen that they introduced. So here are the results, and I'll go through what's being shown for you slowly. So the first thing to note, if we look at the results over here, what they're measuring is what they call uridinyl density. And so uridinia are the asexual spores that are produced by rust fungi. So these are the spores that rusts produce to spread over to different individuals. And the idea here is that they're using this as a measure for how effective the fungal pathogen has been at infecting those trees. And so the idea is that if there's a lot of infection, they'll produce lots of uridinia spores. But if they haven't been able to infect very well, then they'll produce fewer of those spores. Okay, so that's the logic for what they're measuring. I said that they inoculated them into different popular species, and so what they're showing here is the results combined for all the different popular species. Okay, so it's a combination of essentially 6 different experiments, results all shown together. Down here are shown the 4 endophytes, different endophyte species that they used. So these are the results. So these are the results for this particular endophyte species. These are the results averaged across all 6 popular species. Same for this one, same for this one, and same for this one. And then the control is where they haven't inoculated with the endophyte. So this is endophyte-free populus trees. And so what you can see is that there's been a dramatic reduction, particularly for Stachybotrys and Trichoderma endophytes, in the number of uridinia that are produced by the fungal pathogen. There is also some pretty good reduction, but not quite as strong for these other 2 fungal endophyte species. And of course, they're comparing to the control here. So the question that I have if we move to slide number 3 is, what do you think about these results? Is their experimental design good? Are there necessary controls that are missing from this experiment that you need in order to be able to conclude that these fungal endophytes are protective for or provide protection to their host populus tree plants for this fungal rust pathogen? Or from these results, can you simply conclude, yes, it does seem that these fungal endophytes are producing or are providing protection to their host plants against this fungal rust pathogen? So I'm not going to talk about that now. I'm going to leave you to think about that question, and then in the next mini lecture, I'll come back, and at the start of that, I will discuss this point some more. So have a careful think about it, come up with some answers, and then come back and check out how you went in the next mini lecture. So that's all I've got for this one. It's a nice short one. So I'll sign off now. And again, if you have any questions, please post them to Piazza.